Mutation Station.
Thank you for joining the #1 podcast for information about mutations. This episode was produced on March 15, 2020 in Champaign, Illinois. In this podcast, every Monday at 4 p.m., 4 researchers and physicians in the up-and-coming field of mutations discuss what mutations are and the specific mutations that cause the disorders and diseases of today.
Hi everyone, I am Dr. Tivinon and I work at St. Jude Children's Hospital. I'm so excited to talk with you guys about the ins and outs of mutations.
Hi everyone, and I'm Jasmine Lee, a researcher working at the National Institute of Allergy and Infectious Diseases. Hopefully with the knowledge I have, I can give you more of the science behind these mutations.
Hi, my name is Grace Tolentino and I work for the National Institutes of Health. My research focuses on mutations that cause disorders of the immune system. I'm so excited to join this discussion.
Hi, I'm Holly Gussis, a researcher working for Rosalind Franklin School of Medicine, and my research focuses on point mutations and their discovery. I'm glad I could be here to share my knowledge.
So Holly, for all the listeners that don't know, what exactly is a mutation?
Mutations are changes in your DNA sequence which are passed down during cell division. These may be spontaneous, caused by natural biological processes, or they can be induced by mutagens like UV rays, X-rays, and cigarette smoke. Mutations are random events that can have 2 main effects to a gene. It may change the nucleotide base sequence or add or remove nucleotide bases. So Holly, could you tell us a little more about how these mutations work? Do mutations always lead to variation? Good question. In a silent mutation, a single nucleotide is replaced. Since the genetic code is degenerate, sometimes the same amino acid is encoded by the altered codon. For example, AGG and AGA both encode for arginine. So if the last guanine nucleotide was mutated to an adenine, this would have no effect on gene expression. But if the last guanine nucleotide were substituted for a thymine, a different amino acid would be encoded for, affecting gene expression.
What are other examples of mutations where nucleotides are altered in a bigger way?
There are 5 main types of mutations, silent mutations being the first. Missense mutations, the second type of mutation, changes a single amino acid in a polypeptide. Most of the time, this does not have a large effect on protein function, so the gene is expressed correctly. In the case of sickle cell anemia, however, a missense mutation has a very harmful effect on gene expression. A missense mutation in the beta-globin gene turns glutamic acid, which is hydrophilic, into valine, which is hydrophobic. When oxygen concentration is low, this mutation causes hemoglobin subunits to stick to one another, and blood cells turn sickle cell-shaped, causing many negative effects to humans.
Wow, it's crazy how a change in a single amino acid can have such a great effect on humans.
Yes, genes are crazy. The third main type of mutation, nonsense mutations, causes a codon in the DNA sequence to become a stop codon, forming a truncated polypeptide which is very unlikely to function properly. Picture, you're in the middle of a sentence and someone enters
Like this?
Yes, just like that. Nonsense mutations cause a protein not to function properly because its expression was so rudely interrupted by a nucleotide sequence being mutated to produce a stop codon. Frameshift mutations, the fourth main type, are mutations in multiple bases that causes the codons downstream of the mutation to be read in incorrect triplets. If many bases are added or removed, not in a multiple of 3, it causes all of the codons after to be read in an incorrect reading frame, creating a completely inaccurate polypeptide chain. Lastly, the fifth type of mutation, called slipped strand mispairing, forms a loop in the DNA where a repeated codon binds to the wrong repeat pairs, forming a loop in one strand. This forms a completely wrong polypeptide chain. Both of these types of mutations cause the intended gene to fail to be expressed.
Wow, those seem horrible.
Although these mutations seem negative, they are responsible for the great variation we see among species. Mutations over time, through Darwin's theory of natural selection, have selected for traits that allow species to thrive and reproduce. Mutations can be for the worse, in diseases such as sickle cell anemia, but they can also make each and every one of us different in a very significant and beneficial way.
However, just as mutations may have benefits, you are right to worry about some types of mutations that can cause life-threatening disorders. One of which we will talk about today is severe combined immunodeficiency, also known as SCID.
Dang, that sounds serious. Are there any ways for us to know if we have it?
Yes, SCID appears during infancy. Specifically, it's a primary immunodeficiency disease, or PIDD, which affects the immune system by lowering its ability to fight off infections. So if you see your infant with chronic ear, blood, liver infections, you should maybe wonder if your child has SCIDs. It is considered to be the most serious PSS according to the AAAA, or the American Academy of Allergy, Asthma, and Immunology, so it would be best to consult a doctor ASAP in the chances that your child does have SCIDs. What kinds of genetic mutations can lead to a child having SCIDs?
According to the National Center for Biotechnology Information, people with SCID have genetic mutations that lead to defective white blood cells. These cells are part of the immune system, and they help protect the body from pathogens like viruses and bacteria. About half of SCID cases are caused by a mutation on the X chromosome. This mutation occurs in the IL-2RG gene.
What is this gene responsible for?
So, according to Medline Plus from the U.S. National Library of Medicine, this gene codes for proteins called interleukin receptors. These receptors are found on the surface of the cells in bone marrow. The receptors, working with a protein called JAK3 and other proteins, make white blood cells. They also control their growth and maturation. A mutation in the IL-2RG gene would prevent the formation of white blood cells. This type of SCID is called X-linked SCID.
Interesting. But you said only about half of SCID cases are caused by this mutation. What mutations cause the other cases?
Another mutation occurs on the JAK3 gene on chromosome 19, as reported by the National Center for Biotechnology Information. According to Medline Plus, this may result in either a JAK3 protein that doesn't work properly or no JAK3 proteins at all. This mutation would also prevent the formation of white blood cells. This type of SCID is called JAK3 deficient SCID.
Wow, so the interleukin receptors and JAK3 proteins are really important for making white blood cells.
Yes, they really are. But the National Center for Biotechnology Information lists a third mutation that causes SCID that doesn't involve the interleukin receptors or JAK3. This mutation occurs on chromosome 20 in a gene that codes for an enzyme called adenosine deaminase. Medline Plus states that adenosine deaminase breaks down deoxyadenosine, which comes from the breakdown of DNA. Without adenosine deaminase, deoxyadenosine builds up in the cells. Unfortunately, it is toxic to the immature white blood cells and prevents them from maturing. This condition is called adenosine deaminase deficiency, which is another cause of SCID.
These mutations in the white blood cells that Grace has mentioned are what cause the little to no immune response, which furthermore cause serious or life-threatening infections. At the office of the National Institute of Allergy and Infectious Diseases, where I work, the most common infections were viral infections. Pneumonia and chronic diarrhea were the most common result of these viral infections. Isabella, could you tell us some of the ways that you diagnose SCIDs at your hospital?
Well, Jasmine, the diagnosis for SCID is similar to how other diseases are diagnosed, that being having a look over complete medical histories and completing a series of physical examinations on the patient. Blood tests and blood counts are an important part of the diagnostic process.
It's good to know there are capable doctors like you, Isabella, to diagnose SCIDs. Is there any way SCIDs can be detected before any symptoms emerge?
Yes, there is. The advancements of healthcare in recent decades has been a huge benefit to detecting diseases before symptoms arise. For example, for SCID, screenings for newborns are available, and there has been an increasing number of state tests for SCID that are part of a mandated newborn screening process.
What are the benefits of these screenings?
Well, just like many other diseases, infants who have SCID do not show signs of the disease until an infection develops. This is why these screenings are vital to the wellness of a child with SCID. It's better to catch the disease early before it can cause an infection. In today's world, we are doing everything we can to take preventative actions against diseases. It is much like how there are screenings for cancer. It's better to detect cancer in its early stages, for example in its benign stage, before it can reach a malignant stage. So now that we know the symptoms and how it's diagnosed, let's talk about the treatments. I've done a lot of collaborative work with Boston Children's Hospital in finding ways to come up with treatments. The main priority is to prevent infections.
Some ways to prevent infections are by not giving vaccines for your child, keeping your child away from big crowds, and giving your child antibiotics that are recommended by your doctor. Antibody infusions may also be an option since a child with SCIDs does not have the healthy B cells that produce antibodies to fight off infections. Children are able to get subcutaneous infusions, meaning that it is administered under the skin, or intravenous infusions, which are inserted into a vein. Stem cells or bone marrow transplants are the only treatments that could possibly provide a permanent cure.
So, what are the functions of these stem cells?
Good question, Grace. Stem cells are very unique. This is because stem cells have the ability to develop into many different ranges of cells. These stem cells develop into specialized cells such as white blood cells, which can restore the immune functions in a child that is affected by SCIDs. This procedure, of course, comes with complications, but once successful, it can be a life-saving treatment.
Thank you for teaching us about diagnosis and treatments of SCIDs, Dr. Natividad, and to all the experts who joined us this discussion. Well, that's all the time we have for today, so we hope that everyone listening to this Mutation Station episode enjoyed learning about mutations and SCIDs. Thanks for listening.